Hi, everybody. Welcome to our first remote lecture. We're starting a whole new unit, a whole new section of cell biology, so it was luckily kind of a nice breaking point when I saw you guys last. I'm bummed we didn't get to play our games, but hopefully you guys are working on and finishing up those metabolism questions. I've been getting really good questions from people via email, so thanks for engaging with that. And let me know if anything else comes up. So for today, this would have been 2 or so lectures, all put into one presentation. So we're kind of compressing the information in the class just so that I can put out 1 or 2 lectures a week instead of 3 or 4. Okay, so you don't see my lovely face. If you really miss seeing me in person, we can chat about other ways of capturing this lecture. But for now, I figured just going over the PowerPoint I would have presented will suffice. So as we go along, just let me know if there's anything else that comes up. But for now, we're going to talk about protein sorting and vesicle movement. So 2 really important topics, right? Our proteins need to get certain places. We've been talking about membranes, how things are moving through, what processes happen in different parts of the cell. So now we're going to put together how things move where. All right, so our first item, how do proteins get to the right place? All right, so flipases need to be specifically in the Golgi. Those big complexes in the electron transport chain need to be in membranes of chloroplasts and the mitochondria. So how do they get there? And then the mechanism for vesicle movement and fusion. So we know things move from the nucleus all the way through the ER, the Golgi, maybe onto outside the cell, maybe to other parts of the cell. But how do those proteins get packaged in the right spot, and how do they end up in the right place? So thinking about protein movement today. Okay. So if we think about where ribosomes are in the cell, we have our lovely ribosomes here. A lot of them are out in the cytosol, but some of them are embedded in the ER, on the membrane of that rough ER. That's why it's called rough ER. So if we're synthesizing proteins in the cytosol, they're going to end up in certain places. So cytosolic proteins can be put into the peroxisomes, our mitochondria and chloroplasts, or they might get put into the nucleus. Okay, so that's if we have ribosomes out in the cytosol, they're translating these proteins, this is where they're going to end up. Alternatively, if we have a ribosome on the membrane of the ER, we'll talk about this process in a minute, but if they get translated into the lumen of the ER, they can end up in a lot of different places. So they can be transported all the way to the plasma membrane, they can be secreted outside the cell, or they can be put in things like endosomes and lysosomes. Okay, so depending on where the protein is synthesized can determine where it ends up. So today we're going to talk about 3 different modes of how proteins get transported. So like we already mentioned, if we're moving from the cytosol into the ER, mitochondria or chloroplasts, those kind of move in a certain way. When proteins move from the cytosol into the nucleus, there's another kind of special way that things get transported. And then we'll talk about vesicles. And so the big answer to how these proteins know where to go, they'll have some kind of signal sequence that's encoded in the DNA that gets translated and is recognized by a lot of different proteins. So these are usually 15 to 60 amino acids long, and these signal sequences are more often than not removed from the finished protein. So if we were studying these things and we were looking at the DNA of one of these proteins and we compared it to the final amino acid sequence of this functional protein, that end protein would actually be a little shorter, a little smaller, because that signal sequence was taken off. Okay, so we'll revisit these 3 types of movement in our lecture today. Okay, so starting at the top of the page here, ribosomal subunits cycle between bound and free. So it's not like when a ribosome gets made, it's directed one of two places. Okay, you go to the cytosol, you go to the ER, and that's where you stay. Ribosomes get made, and they kind of are just floating around. So if one attaches to an mRNA, it's making this protein, it's going to stay in the cytosol. So sometimes we can get multiple ribosomes binding at the same time, making lots of proteins simultaneously. That's this polyribosome we're seeing in action right here. So it comes out, there's nothing to recognize, it's just going to stay in the cytosol. All right, when we're done translating this protein, ribosomes dissociate. They're free to just kind of mingle. And then they're going to recognize this mRNA. It's going to start translating this ER signal sequence, so the endoplasmic reticulum signal sequence. This is actually going to orient it to a protein in the membrane of the ER. So this mRNA is translating the signal sequence. So this amino acid sequence is going to be recognized by a protein in the membrane of the ER. So this is all going to localize to the ER membrane, where it's going to continue translating this protein. So this signal sequence is actually going to bind inside this protein right here. And so you can see as we're translating it, it's actually going into the ER lumen. So rather than making a protein fully on the outside and then importing it in, the ribosome is bound, and as it's translating, it's adding these amino acids. It's putting this protein directly into the ER lumen. So ribosomes cycling around. As soon as this protein is finished translating, it can dissociate and go back to the pool. So again, it's the signal sequence on the mRNA itself that gets translated that brings the ribosome and the RNA to the ER. So here's a more in-depth view of how that works. So that signal sequence is going to be translated. It's recognized by the signal recognition particle, or SRP. So this SRP protein is the thing that actually brings it to an SRP receptor. These are embedded in the membrane. Once these are associated, a protein translocator, so this protein that spans the membrane, is going to recognize this ER signal sequence right here. The SRP is dissociated. The sequence is able to bind to the inside of this translocator, and we're able to bring this newly forming protein into the ER lumen. So again, rather than fully forming a protein and importing the whole thing, the way we bring proteins into the ER is as they're actively being made. So we're translating these proteins directly into the ER. Often there's going to be chaperone proteins to help it fold into the correct shape that will be in this region. So again, signal sequence recognized by the SRP. SRP binds this receptor. Once this recognition event takes place, that SRP can be displaced. ER signal sequence binds to the translocator, and we're able to bring in our protein. Okay, so if you remember from our hydrophobicity plots, right, sometimes we have proteins that don't come all the way into the ER lumen. If we want things that are going to be bound to a membrane, we want them to stay in some kind of membrane. And so there's three different kinds of proteins that are going to be coming in. Soluble, things that are going to come into the ER, probably be transported elsewhere. Single pass transmembrane proteins. So single pass, there's just going to be one domain that's in the middle of the membrane. as well as multipass. So soluble, they're going to come all the way in, single pass and multipass transmembrane protein. So here's that first one we were talking about. So that signal sequence finds our translocator. As we're elongating our polypeptide chain, it's going to continuously come in. At some point, a protein called a signal peptidase will cleave the signal peptide away from the rest of the protein. That channel will close, and then we'll have our fully formed protein in the ER. That signal sequence will kind of stay in the membrane to be degraded, so those amino acids can be used for other things. So once our protein comes all the way through, our signal peptidase is able to cleave that sequence, close that channel, and now we have our mature protein that we can send off elsewhere. If we want something to stick in the membrane, we need another signal to make that work. So in this case, we have a single pass transmembrane protein. We're going to add something called a stop transfer sequence. So these are usually going to be pretty hydrophobic, right? We want them to stay in the membrane. So we have our original signal sequence. This is our start transfer. This orange one will be our stop transfer. So like we've mentioned, this is going to bind that translocator. We're going to keep bringing in this newly synthesized polypeptide chain. However, once the stop transfer sequence makes its way into the channel, it's going to signal this peptidase to cleave that start signal, and we're going to stop movement of this protein. So just like it says, stop transfer sequence stops bringing in that protein, and we're left with this membrane domain with this part of the protein on one side and the N-terminus on the side. For multipass, instead of having our signal sequence at the very beginning of the polypeptide chain, it will be somewhere more in the middle. So in this case, our N-terminus is going to stay on this cytosolic side. This is going to bind our translocator. It's going to tell us to keep importing this polypeptide chain, but it will stop once we get to the stop sequence, and then our translocator will close. So we've been able to add 2. So we've seen that some proteins have multiple membrane-spanning domains. So that's just going to mean there's going to be a combination of these start and stop transfer sequences. Okay, so it's not only are these hydrophobic regions important to stay in the membrane, some of them also have signal sequences that direct that protein movement and direct how they're shaped. So that was movement into the ER. Once proteins get here, often they are modified by adding these big polysaccharide sugars right here. So often when we bring proteins into the ER, they'll be converted to glycoproteins, so sugar proteins. These sugar groups do a couple of things. So one, they might provide protection from degradation while they're in the ER. So when we see lots of polysaccharides, think signaling. So one can signal, don't break me down, I'm going to go do something else. And another function is that they can act as a signal if we're putting this protein on the cell surface. So on the cell surface, often we have something called the glycocalyx, so kind of a sugar coating on the outside. And the makeup of these polysaccharides act as signals. They help with cell-cell signaling. and those often will end up on the extracellular side of the membrane. So if we need to add any of these polysaccharides to an incoming protein, there's a certain kind of glycolipid that helps out, which is called dolichol. So this is a lipid that's membrane bound, has these phosphate groups that attach to a really big polysaccharide molecule over here. So instead of transferring individual sugar molecules onto a protein, it's going to transfer this whole complex in one step. And so this big transfer of this oligosaccharide is going to occur with our oligosaccharyl transferase. So this is our enzyme that's going to catalyze this, putting this big oligosaccharide from our dolichol onto our growing protein. And more often than not, they're put on the asparagine residues of these proteins. And again, addition of these sugars acts in protection and also signaling as it's going through. So that was into the ER. When we bring things into the endoplasmic reticulum, they're still being translated. So as they're being made, as those proteins are being put together, they're entering the cytosol of the ER. So kind of similar recognition mechanism for mitochondria and chloroplasts. However, instead of still being bound to the ribosome, these proteins, these precursors over here, have some kind of signal sequence. This sequence is going to bind to an import receptor. And because we have this double membrane in these organelles, we have a translocator in the outer and in the inner membrane. So wherever these import receptors are, they'll bind this recognition sequence and they'll move to that corresponding inner protein translocator. So a protein has bound, it's going to move. The signal sequence is also going to recognize this protein translocator on the inner membrane. So we actually have to unfold this precursor protein to bring it in. So we've translated it. It might have folded into some kind of shape. It's going to bind these receptors. It's going to be brought in. And as it's being brought in, it needs to unfold to fit through this channel, through these translocators. So at some point, it's going to come all the way through into the matrix, where there will be other proteins that are able to cleave these signals. There might be other chaperone proteins that help put this protein into its mature form. So again, these have to be unfolded in order to enter the mitochondria and the chloroplast. So that was all that first kind of movement we mentioned from the three that we're going to talk about today. And so now we're going to look at how things are brought into the nucleus and how things are taken out of the nucleus. So we mentioned nuclei way back at the beginning of the semester. The way things move in and out is through that nuclear pore. And so this image in the middle is our cartoon of what that nuclear pore actually looks like. Instead of being a big hole, it's actually this complex of different proteins, I think about 30 or so, that have these little fibrils in the middle. This forms a sort of mesh. Your book likens it to a kelp forest, some kind of not super dense, but something that fills the space here that lets smaller molecules through. These diffuse just fine, but prevents a barrier of entry for larger molecules. So not that they won't be able to make their way in, but they need to enter through some kind of active transport. So this mesh provides the structure that lets in small molecules but keeps bigger ones out. So when scientists study these, they can isolate nuclear pores and kind of put them all together. And when we look under the microscope, this is their shape. So we can see all these different proteins that are in the membrane, and then this middle section where we have that mesh that prevents everything from getting through, that prevents certain things from getting through. So if you think about what needs to go into the nucleus and what needs to come out. So we know certain things are made in the nucleus, those need to be exported, but in order to do functions like DNA replication, we need specific enzymes, and since they're synthesized outside of the nucleus, we need to bring them in. So this next slide is going to walk us through different things that move in and out of the nucleus. So here's our nucleus right here. We have our nuclear pores with that mesh in the middle that prevents bigger molecules from getting through. So if we follow these red arrows, these are the things that we're exporting. Right? So our DNA isn't leaving, but we need to let the mRNA leave to get translated. tRNAs and rRNAs, all of these RNAs that are transcribed that need to leave to function. So these can be part of our ribosomal subunits. They'll be exported. mRNA needs to leave. tRNAs, all contributing to translation. And then once we've made certain proteins, right, these completed proteins, maybe we've made a DNA primase, maybe we've made a DNA ligase, those would need to go back into the nucleus to help with DNA replication. Maybe we've made some histone proteins, those need to go back into the nucleus. Things for transcription, etc. So these are the kinds of things that need to be moving in and out. So now we're going to see that mechanism for how they're able to do that. So focusing on the diagram for now, we've been looking at a lot of enzymes that rely on ATP. Now we're going to focus on molecules that rely on GTP. So the way we get things inside and outside of the nucleus, this process is regulated by a molecule called Ran. So Ran is a GTPase. And so just like that ATPase, it binds ATP, and other things can take that phosphate off, so it can bind GDP, or ATP, excuse me. ATPases bind ATP. Other things will extract that phosphate so it can be bound to ADP. Other proteins can put a phosphate back on. So GTPases are the same thing, but they're binding GTP instead of ATP. So in any case, we have our Ran GTP in the nucleus. This molecule can leave the nucleus. It's going to encounter this Ran gap protein. So gap stands for GTPase activating protein. So our Ran GTP leaves the nucleus, encounters this gap, which takes off a phosphate. Now we've activated it. So Ran GDP is able to bind another molecule. Once it does, it can come into the nucleus, where it will interact with this Ran GEF. And GEF stands for guanine nucleotide exchange factor. So this is just going to exchange a GDP for a GTP, and we can start the cycle all over again. So GTPase activating protein is going to remove a phosphate. Guanine nucleotide exchange factor is going to add that GTP. Okay, so we'll see how this molecule plays a role in import and export in the next slide. But just so that we know that this molecule is cycling through having that phosphate taken off, having GTP put back on. So here we're looking at how we're bringing something into the cell. So export's going to look very similar. We're going to start with import for now. So let's focus at the top of this diagram here. So we have some kind of protein. We want to bring it into the nucleus, and we know we want to do that because this protein has a nuclear localization signal. So it almost looks like a Pringle in this example. But this kind of dark area right here is our localization signal. We're going counterclockwise to this diagram, so we're going to follow the arrows this way. So our protein we want to bring into the nucleus is going to be recognized by this nuclear import receptor. So this import receptor binds our protein. It's able to come into the nucleus through that pore. It's going to encounter our molecule, our Ran GTP. When Ran GTP binds our import receptor, it lets go of that protein. In this case, it's our cargo. So we've delivered our protein into the nucleus. This import receptor is still bound to Ran GTP. It can now leave through that nuclear pore. It's going to encounter that gap, that activating protein, which will remove a phosphate that will dissociate the Ran, now GDP, from that receptor. So now this import receptor is available again to bring in another protein. So these import receptors are able to move through the nuclear pore. So that's why it can either transport in our cargo protein or bring Ran GTP out of the nucleus. And this description on here is just in addition to walking through this diagram over here. Okay, so that was nuclear import. Again, export's going to be a very similar process. So we still have our Ran GTP in the nucleus, our Ran GDP in the cytosol. So in this case, we're going to start at the bottom of this diagram. We have something inside the nucleus we want to bring out. So earlier we had an import signal. These proteins are going to have an export signal. It's going to bind our export receptor, which is already bound to Ran GTP. So this export receptor with our cargo in tow and our Ran GTP is able to move through that pore. It's going to encounter that molecule that dissociates that phosphate, that gap. So that GDP will dissociate, that Ran GDP will dissociate, delivers this protein to the cytosol, and now this export receptor is free to move back through the pore to bind another Ran GTP and another protein that needs to leave. So again, the process of hydrolyzing that GTP is what allows the movement, that release of our cargo protein, and that the Ran GTP is what's able to move our proteins through that pore. So just so that we can have them both side by side, here's import and export right next to each other, and we've added the function of our enzymes here. So when we're importing something, we bind our nuclear import receptor. It's able to get brought into the nucleus. Ran GTP binds, dissociates our cargo. we're able to move back through. It encounters gaps, which phosphorylizes the GTP, releases that RAN, and now we're able to bind a new protein. So RNA can also be brought through. RNA protein complexes can also be brought through this way. So bound to this export receptor, which is also bound to GTP, coming through here, encountering that gap, releasing our protein RNA protein, able to come back through. Okay, so when we saw with the ER process, with chloroplasts and mitochondria, those had to be unfolded or they were being made as they were being brought in. This mechanism allows proteins to come in or leave fully formed into and out of the nucleus. So that third way we talked about movement is through vesicles. So often when things are brought into the ER, they're going to be transported far and wide thanks to the movement of vesicles. So if you want to see an overview of this process in motion, feel free to pause this video and go check out this virtual cell video of vesicle movement. It moves things through the ER, through that Golgi network, and on through. So again, feel free to pause this, take a look at that, and then come back. I know seeing things in movement helps solidify the ideas a little bit more. Okay, so we're going to switch to a pen here. So as we're looking on the left side of the diagram, we have our nucleus, we have our ER, and if we follow the arrows at the top of the diagram going this way, we're looking at our secretory pathway. So things that we're exporting, also called the secretory. Secretory pathway brings things out of the cell. All right, so we're going to go through here. Vesicles are going to move it through the Golgi. These secretory vesicles then can bring them to lots of different places, but often to the outside, these vesicles will fuse with the cell membrane, and the contents then can be released. But vesicles also function in the opposite direction. So we call that the endocytic pathway. So endocytosis involves endosomes. The membrane will invaginate right here at the cell membrane. It'll form these vesicles. And these then can fuse to the Golgi, make its way into the ER. So as we're looking at the top of the diagram, we're following the secretory pathway. As we're following the bottom of the diagram, we're following the endocytic pathway. So retrograde transport just means instead of the canonical movement from the inside to the outside of the cell, we're moving proteins and other cargo back this direction. So yeah, check out this video if you want to see this in action. But we're going to talk about the actual mechanism of how these vesicles are filled with the right types of proteins and how they're able to move through, how they're able to bud, and how they're able to fuse. So here's just our cartoon conception of how we're making vesicles and how they're moving through. So some kind of donor compartment, if that's the ER or the Golgi or our endosome. We have some kind of lumen with our proteins, our soluble proteins here, or our membrane proteins here. It'll go through this budding process. We'll have a mechanism for pinching off this piece right here so that it's able to move this way. And instead of just kind of floating through, more often than not, these vesicles are attached to motor proteins that are crawling them along the cytoskeleton. We'll be chatting about the cytoskeleton next week or very soon. Okay, so we've pinched off a vesicle. It's able to travel along this network. Eventually, we need it to fuse to the target compartment. So it might be the outer cell membrane, it might be another organelle. But in any case, we need to, some process to bring these two membranes together, to let in what's inside the vesicle and possibly also to transport these membrane proteins. So let's see how that works. All vesicles are going to have some kind of protein coat that helps them bud from that donor compartment. So the most well-studied coats are these clathrin coats right here. So let's check out how we form a vesicle. So we have some kind of cargo molecule. It might be a protein that needs to go, it might be a hormone that needs to be secreted from a cell. It could be lots of different things. They're going to bind to a very specific cargo receptor. So that's this recognition process for how we package the right thing. So our cargo binds to this receptor. These different receptors can bind to a molecule called adaptin. So that's this light green molecule right here. There are quite a few different adaptins, but they can bind many different cargo receptors. So this is that specific interaction. They're able to bind to these adaptins. This adaptin then binds to our clathrin. So this dark green protein right here. So once we have enough cargo molecules binding to these receptors that are then bound to these adaptins, we're recruiting these clathrin molecules that form this mesh over here. So this coat is actually forming a structure that encourages this vesicle to form. So the more adaptins, the more receptor molecules we have right here, the more the membrane is going to curve around. So the shape of these clathrin coats are what help shape those vesicles. Once we get to the point where we need to pinch off this vesicle right here, we have a protein called dynamin that's going to come in, and its shape helps encourage the budding off, the split of this membrane right here. So then that clathrin coat encourages vesicle formation. Dynamin comes in and helps pinch that vesicle off from the rest of the membrane. Once the vesicle is released from that donor compartment, they're going to get rid of this coat. So this isn't necessary for vesicle movement. In fact, sometimes we want our vesicle membrane to be bare so it can be recognized by different things. So this coat then is going to come off, and our vesicle is able to move through the cell. So clathrin-coated vesicles are the most well-studied, but there are lots of other coat proteins that help the formation of vesicles. So depending on where you need to go as a vesicle will kind of determine what coat you're going to wear. So if it's really cold outside, you want a heavier coat. when vesicles are concerned, depending on where you're going, you're going to want a different type of protein to help you get there. So COP just stands for coat protein. So clathrin was the most creatively named, but now people are looking into these other coat proteins. So we know coat protein 1, COP2, designated by these blue dots or these red dots. So as we're leaving the ER, we're most likely going to make vesicles with these COP2 proteins. They're able to fuse with the Golgi. As we're leaving the Golgi, we'll probably form COP1, coated vesicles. If we need to leave the Golgi, or if we need to come in from the cell in that endosome pathway, we'll be using these clathrin coats. So again, those coat proteins help vesicles form, and then they're removed for vesicle transport. So the way, well, make sure you double check things before you submit them. But the way we're going to then fuse a vesicle to the membrane has a couple different recognition steps. All right, so we don't want to mail something to the wrong address. We've packaged our cargo protein really nicely. We want to make sure it gets to the right spot. And so vesicles are going to have some kind of Rab protein. These are another example of GTPases. But all we need to know for this mechanism is that this Rab protein is going to act as our address label in a way. So Rab is going to be recognized by something called a tethering protein. And this tethering protein is attached to the membrane we want to fuse with. It's going to recognize that Rab protein. So if these didn't recognize each other, it wouldn't stick and the vesicle would move along. But because this is our end destination, this Rab protein is going to affix to this tethering protein, and we'll be able to bring that cargo into the membrane. So this first step is our tethering step, and that's just that recognition of Rab to our tether right here. Okay, so we also see these snare proteins. These will come in a second. So first step is tethering, the recognition of that tether with that Rab protein. The next step is docking. Right, when we bring our boat in to the dock, when we charge our phone on a docking station, we're making physical contact with our vesicle, with where we want to bring it in. So this docking step is going to be facilitated by these snare proteins. So our V snare is this red squiggle right here. V stands for vesicle. The snare protein is going to interact with a T snare, named for the target. So our T snare is going to recognize our V snare for vesicle, and it's able to bring the vesicle in really close to the membrane. This process of fusion takes a lot of energy. It's a very unfavorable reaction. We're trying to force two hydrophobic things together. And so the way we do that is these snare molecules will twist and physically pull those membranes together. So the point I want to make here is there's 3 steps to getting vesicles to fuse to its final location. It relies on this tethering or recognition between the Rab and the tether, the docking process of these snares recognizing each other, and then fusion is facilitated by these snare proteins wrapping around and getting that membrane to fuse. Then our proteins can be released. So we'll see that docking and fusion step in action. So we don't have them pictured, but our Rab protein and our tethering protein, right, are in contact. We know we're in the right place. Our V snare molecules are going to interact with the T snares. The vesicle has docked. Once these have interacted with both, right, they're not going to move anywhere else. Slowly, these two snares are going to twist around each other, and as they twist, they're pulling kind of in opposite directions, so they're bringing these membranes closer and closer together. After a certain amount of twisting together, those membranes will fuse. So it's the action of these protein interactions that allow that membrane to fuse and allow our cargo to enter the target compartment. So that was looking at how proteins are moving around the cell, how we can put specific proteins in the right place. We talked about movement into the ER and into things like mitochondria and chloroplasts. We also talked about the specific mechanisms for nuclear movement, either import or export, and we looked at how vesicles are able to move around the cell. So keep working on those metabolism questions. I've gotten a lot of good questions over email, so wonderful to see you guys thinking about those. Skim pages 525 to 551 for a lecture I'll post early next week. So like I said in my email, my goal is to upload either 1 or 2 lectures at the beginning of the week. So goal is on Mondays. Then you'll have some kind of, what's the word? Like a quiz, a comprehension assessment for each lecture. So we're replacing exam 3 with these comprehension quizzes. And we don't have details on what finals will look like yet, but we'll use those quizzes in the meantime. No quiz over this lecture for this week since I'm getting it up on Friday, but this material will still be important. So some of this might show up on a quiz next week. We had on our schedule doing an ELISA lab activity next week on Monday. I found a really nice virtual lab activity, so more information from me about that on Monday. I'm still looking at other activities we can do for the other 2 labs that we had scheduled. But no quiz for this lecture, although don't forget all this information. Metabolism questions due. Don't forget about your literature presentations. You'll get more details from me about how we'll record those. And then we have another activity that I'm going to talk about next week. So stay tuned for more announcements. Otherwise, keep working on those metabolism questions. Again, feel free to send any comments. Specific feedback is really helpful, especially if there's something you'd like to see more of or see something different as we're making this big transition. So thanks for your attention. Go ahead and get started skimming cell signaling information for next week. I'll give you guys an updated schedule as I finish putting that together. All right, thank you, happy studying, and you'll see more from me on Monday.